Good afternoon, everyone. Thanks a lot for coming to today's FENS Friday webinar. Today we will be dealing with neuroanatomical tract-tracing techniques. We're getting started with classical things and going to more modern approaches. So the seminar will be recorded and will be later on available at the FENS website. The seminar is the webinar. Oh, sorry, I need to share my screen. Sorry. I think it's that one. Okay, so you can see now my screen. So welcome again to this FENS Friday webinar. Today we will be dealing with neuroanatomical tract-tracing methods. This webinar is going to be made of, it's going to be made of 3 different presentations. Today we have 2 wonderful panelists, Anne Bieler from Bordeaux in France, and Nicolas Foster from UCLA in California. Nicolas woke up very early today, so sorry, Nicolas, for waking you up so early today. It should be somewhere around 5 a.m. in California. The overall idea is that for every presentation, these are not going to last for more than 15 minutes in order to allow to have later on at least 15 minutes for Q&A session. So during the talk, if you have any question, please use the chat. And later on, once all the talk are finished, we will go to the chat trying to answer one question after the other. So my role here is just to introduce the overall topic. You know, the brain is a very complex organ. It's made out of circuits that are quite sophisticated. So, you know, the overall idea of this seminar is to provide attendees with some tips and tricks based on our own experience. So at the end of the day, I will be a very happy guy if people can elucidate which tracer or which combination of tracer are better suited to solve a given experimental paradigm. So choosing a given tracer is not an easy choice, but you know we will try to do our best in providing you with the underlying rationale behind these techniques. What can be done with tracers that is quite impressive, particularly with the new arrivals that Nicolas and Anne will be dealing with. So, you know, in principle, it's quite very easy to provide you with, let's say, a definition of tracers. There are substances that are, are injected in the brain, and once injected in the brain, are taken up by neuronal processes. And tracing can be, performed in 2 different ways. So when the substance that has been injected is taken up by cell bodies and transported all the way down to the axons until the terminals, this is called anterograde transport. And things that behave this way are called anterograde tracers. However, transport can be performed into the opposite direction. So, once injected to the brain, the neurons that innervate the injection site can take up these tracers on the terminals, and it's going to be transported back to the parent cell bodies, and these are called retrograde tracers. However, you should also be aware that there are a number of tracers that are transported in both directions, and this is what we call bidirectional tracers. So, in an attempt to provide you with, let's say, a landscape view of, the history behind tracing, as you can see, it's quite a rather short history, beginning 70 years ago, at the beginning of the 50s, with the silver degeneration techniques. And later on in the 70s, it became horseradish peroxidase, a number of fluorescent tracers. Entering the 80s, the arrival of cholera toxin, that you will be hearing quite a lot about cholera toxin today. Fluorogold is a wonderful retrograde tracer, together with PHEL, introduced in 1984 by Gerfen and Shevchenko. At that time, it was the only anterograde tracer available. This, really shake the ground, or really shake the field. Later on, we witnessed the arrival of a number of neurotropic viruses that most of them are no longer in use. in the 1990, was the arrival of bioattenuated dextranamine. It's absolutely wonderful anterograde tracer. And now we are here. We are in the era of a second generation of viruses. At least in my view, the arrival of cholera toxin and fluorogol as two wonderful retrograde tracers. This really changed the field. And in the field of anterograde tracing, you need to keep an eye on PHAL and on bioattenuated dextranamine. They are the two very good ones as anterograde tracers. So just to compare, a number of properties of one tracer versus the other, for cholera toxin, just a few words. It's a very good retrograde tracer. We have a primary antibody against cholera toxin that is superb. A number of fluorescent conjugates have also been made available more frequently. You can deliver CTB either by pressure or by iontophoresis, depending on what you want to do. For instance, in my work in monkeys, I found that cholera toxin is particularly well suitable for performing large injections. In monkeys, you have big nuclei that you need to fulfill with the tracer. To some extent, it's a bidirectional tracer. And so far, to the very best of my knowledge, performs always very reliable in any animal species you may have in mind. Compared to fluorogol, fluorogol is also a very good retrograde tracer, with also a very good primary antibody in detecting fluorogol, although fluorogol is fluorescent in itself, with a very strong signal. So fluorogol is very resistant to fading. You can deliver fluorogol either by pressure or by iontophoresis. And at least in our hand, what is critical is to dissolve fluorogol in cacodylac buffer. Perform much better in cacodylac buffer when compared to a phosphate buffer or something like this. It's a pure 100% retrograde tracer. And some people will express a number of concerns when using fluorogol in macaques. In our hands, it works pretty well. There are people that may claim that fluorogol is not 100% reliable in non-human primates. For anterograde tracers, you have two main choices. Bioattenuated dextranamine, BDA, introduced in 1990 by Wieman and Tony Rayner as a very good anterograde tracer. One of the main advantages of BDA is that the tracer in itself is already bioattenuated, meaning that there's no need of using antibodies to stain for BDA. And this property makes BDA particularly well suitable when used in combination with several other tracers, because you don't have any problem in terms of cross-reactivity of antibodies. It's a bidirectional tracer. You may expect some degree of retrograde transport of BDA, particularly when delivered by pressure. There is another close relative to BDA with a smaller molecular weight, that is BDA of 3 kilodaltons. And BDA is very good for ultrastructural studies. Compared to PHEL, PHEL is 100% anterograde tracer. It doesn't work always the same. There are people that may claim that has to some extent capricious in terms of performance. Can only be delivered by iontophoresis. And in our hands is the typical second choice anterograde tracer. However, you need to keep in mind that brain, in terms of circuitry, is quite a very complex organ. So most of the time, there's not too much you can do with just one single tracer. So in most cases, nowadays, we are performing multiple tracing paradigms, combining at least 2 or 3 tracers or combined with something else. So there are a number of, let's say, golden rules when selecting tracers for multiple tracing paradigms. For instance, wherever possible, you need to combine tracers that are transported in just one direction, either anterograde or retrograde. And it's also, I would suggest to use tracers that can be combined together, but tracers with a similar nature. So, just try to avoid combining non-fluorescent with fluorescent tracers. Another important issue is that the tracers to be used in combination should afford for a similar survival time. For instance, there are tracers that can be, perform quite pretty well with a survival time of just 1 week. However, there are a number of tracers with a survival time of less than a couple of days. So, my point is that when you are about to choose tracers, wherever possible, use tracers with a similar survival time, just to avoid repeated surgical procedures. Of course, you need specific primary antibodies and secondary antibodies without any type of cross-reaction. And in the ideal world, the tracers you're about to use, it's going to be nice if these tracers can be detected both by immunoperoxidase and immunofluorescent techniques. So, the advantage here is that most of the people will rely on multiple fluorescent paradigm. That is okay. However, if you want to have some permanent stains, you better try to convert these signals to an immunoperoxidase stain. And wherever possible, for people going very deep into the tissue with electron microscopy, you need to use tracers that can be detected with electron microscopy. Okay, so I'm going to show you just a few examples of what can be done with multiple tracing paradigms. So in this image, you have a section of the substantia nigra of the monkey brain with a triple staining paradigm. Here we have combined retrograde tracing with chloratoxin that is located here. These brown neurons are neurons in the substantia nigra pars compacta projecting to the putamen nucleus. And what you have here in purple, staying with a peroxidase chromogen called VIP, are neurons projecting to the caudate nucleus. So this is a dual tracing paradigm. These neurons are receiving fibers coming from the internal division of the globus pallidus, has been labeled with BDA. Okay, it's a typical example of triple immunoperoxidase stain with 3 different chromogens, DAB, nickel-enhanced DAB, and VIP from vector. This is the fluorescent correlate. And these images were taken from the rodent striatum, where we are being combining chloratoxin that has been injected in the internal part of the globus pallidus with fluorogold that has been injected in the external part of the globus pallidus. So here we can disclose neurons giving rise to either the indirect or to the direct basal ganglia output pathways. These neurons receiving fibers coming from the thalamus through the thalamus striatal pathway that has been identified upon the delivery of BDA in the parafascicular nucleus of the thalamus. This is another example. In that case, we are combining retrograde tracing with Rabies virus, these neurons in green, where you can see all the dendrites. These are medium-sized spiny neurons in the rodent striatum. You can even see the spines in the dendrites. And these neurons are surrounded by 2 different types of fibers. Those in blue are coming from the cortex and are being labeled with BDA. And those in red are thalamus striatal fibers coming from the parafascicular nucleus that are being labeled with PHAL. This is another example of rabies virus. You need to be aware that rabies virus is a transsynaptic vector. So depending on how long you wait after the injection of the rabies virus, if you wait long enough, you can see second-order neurons by transsynaptic transport, meaning that neurons projecting to the primary order neuron, to the first-order neurons, can be transsynaptically labeled with rabies virus. So this is an example of thalamus striatal fibers coming to the striatum in rodents labeled with PDA, immunohistochemistry for choline acetyltransferase to disclose cholinergic neurons into the striatum, and immunostain for rabies virus. So you can see a typical medium-sized spiny neuron. And what is nice in this picture is that from the neurons that are being stained as positive for choline acetyltransferase, only one of those neurons is secondarily transduced with the rabies virus, meaning that this particular neuron here, you can see with the arrowhead, we know for sure that these neurons are innervating striatofugal neurons, and not this other one that remains only positive for choline acetyltransferase. This is another example of multiple immunoperoxidase stain. This picture was taken at the level of the entorhinal cortex of rats, where we have 3 different things here. We have BDA fibers going from the perirhinal cortex to the entorhinal cortex. We have fluorogold positive neurons that are projected to the dentate gyrus. So we have injected fluorogold in the dentate gyrus of these animals, and we have able to identify entorhinal neurons projecting to the dentate gyrus through the perforant path. And what you have here in purple is a parvalbumin positive interneuron into the entorhinal cortex being innervated by BDA positive fibers. And this is the ultrastructural correlate. Here you have axons labeled with PDA. This is the typical flocculent precipitate of DAB at the structural level. These axons making a synapse into the dendrites of parvalbumin positive neurons. And this is the typical, this type of granules are the typical ultrastructural correlate of the chromogen, of the chromogen that is called VIP. Further on, you also need to be aware that sometimes you need a functional correlate of your circuit. I mean, you can identify a given circuit, and then you may be interested in elucidating the type of, in that case, mRNAs being expressed by these projection neurons. So here we have 3 neurons in the internal division of the globus pallidus that has been labeled with cholera-toxin injected in the thalamus. So these are basal ganglia output neurons. And in that case, we have combined the immunofluorescent detection of cholera-toxin with dual fluorescent in situ hybridization for GAD65 and VGLUT2. So with this type of paradigm, you can disclose, basal ganglia output neurons expressing both mRNAs of different sources, GAD65 and VGLUT, in the same neurons that have been formally identified by retrograde tracing. This is another example of a functional correlate of a tracing experiment. Here we took advantage of the retrograde spread of BDA. So we have injected BDA into either the internal or external division of the globus pallidus. And these neurons belong to the putamen. So these are basal ganglia output neurons from the putamen, from the direct pathway and from the indirect pathway, innervating either the internal or external division of the globus pallidus. With the neurons, we have combined it with a technique called in situ proximity ligation assay, PLA. And in that case, disclosing a pair of GPCR receptors that are D1 and D2. So you can, every single red dot is a pair of D1 and D2 receptors identified within putaminal output neurons. So I will try to end my talk here. For those of you that might be interested in going to deeper details, I have just, shown here a couple of review papers I did with my former mentor, Rabies Virus in Amsterdam. There is a classical review in 2011 and another update in 2020. So for those of you who want to go deeper on into more details, I will strongly suggest it to take this paper as a reference to get started with. And that's all on my side. So now I'm going to leave the floor to Anne. She's going to present some tips and tricks on tracing based on her own experience. So thanks a lot.
Hi, everyone. Can you hear me and see my screen? Excellent. Okay, so thank you very much. Thank you also for the invitation to share some of the principles and some examples of viral tracing approaches using classical histological techniques or whole brain imaging. So as for tracers, viral vectors can be anterograde and retrograde. Currently, the most used anterograde vectors are adeno-associated viruses, or AAV, that can come into 9 different flavors, or so-called serotypes. And today, there are also hybrid serotypes between these different versions. So the virologists have characterized the molecular properties of these viruses, and you can find very interesting papers on that. But what interests us as neuroscientists is how this affects efficacy and tropism of the viral vectors. And as you can imagine, it does change them. So here you have an example of an interesting study. So with identical injections of viral vectors with the same titer in the hippocampus, you can see that just changing the serotype will dramatically change the expression pattern. You also have to take into account that depending on the brain structure and the cell type or the circuit that you target, you might have different efficacy for the same serotype. So for example, here AAV5 in the hippocampus has a very strong expression, and here in the somatosensory cortex, it's much less expressed. So Retrograde vectors on the other side also have different efficacy and tropism, and there is many of them that are currently used. So retro-AV rabies virus that we just heard about and will hear about in the next talk, canine adenoviruses or CAV, and herpes simplex viruses or HSVs. And here's an interesting study that developed the retro-AAVs, and from in their hand, the most efficacious retrograde vector or tracer is the retro-AAV. It's interesting to note also that anterograde viral vectors also have a tiny amount of retrograde transduction. So it's important to keep these in mind when you choose your tracer. Okay, so today I'm going to share 3 examples of experiment based on viral approaches. In the first one, we're going to see anterograde mapping of all projection of a specific brain region, then mapping of selective projection and their collaterals, and finally, the use of whole brain mapping for selective projections. So for the mapping of all the projection of one brain region, I'm going to use the example data from my lab on the anterior and posterior insular cortex. So for this experiment, we injected AAV of serotype 9, carrying the gene coding for EYFP under the control of the CAMK2 promoter, which will lead to expression in glutamatergic excitatory neurons. Then 3 to 4 weeks after injection, we will extract and section the brain in 100 micron slices, and then image target regions. So in this case, we choose 12 target regions using a confocal microscope. So here you have the example of these 12 regions for the injection into the anterior insular cortex. And as you can see, the brightest axonal terminals are in the basolateral amygdala, BLA, and contralateral anterior insular cortex. Interestingly, when you look at the posterior insula, you find a different pattern with the highest density in the central amygdala with the medial and lateral part, and the contralateral posterior insular cortex. So this is just 2 examples. You can quantify across different animals. So this is the result for 8 different animals. So you quantify the intensity of fluorescence across these images, and we represent them as their relative density compared to the region that has the highest density. So here you see the highest is in the BLA, and here in the central lateral amygdala. I'm not going to go into details. The idea is that you can apply this technique to your own favorite circuit. We like to represent the data in using these pie chart where the thickness of each line represents the density of the relative density of the projection. And then you can compare the projection from different regions. So here is the anterior and posterior insula, but you can use that for any brain region. So here, for example, you can see that for the BLA, the densest projection is from the anterior insula, not the posterior, and in the contralateral anterior insula, it's 90% projection from the anterior insula. Okay, so for the second example, what if you want to map a selective projection from one region A to a region B? So in this case, I'm going to use the example of 3 selective projections from the basolateral amygdala. And in this case, to be really specific of these neurons, we use a dual viral strategy. So in the downstream region here, the example is with the nucleus accumbens, we inject the retrograde CAF2-CRE virus. So it's expressing the CRE recombinase, and it's going to transduce retrogradely the neurons, so to the BLA and all region projecting to the nucleus accumbens. And then in the upstream region, so the BLA for us, we injected the AAV5 viral vector carrying the gene coding for channelrhodopsin EYFP. For the anatomy, what interests us is the EYFP aspect. And this is in a double inverted open reading frame, meaning that when the CRE recombinase meets this viral vector, you have the flux site that allows the flipping of the gene coding for EYFP, and then you will have expression. So that means you will have fluorescence only in the neurons projecting from your region of interest to the downstream target where you injected the CAF2-CRE. So we did that for different projection to the central amygdala and the ventral hippocampus as well. And again, after 6 weeks of expression in that case, we sliced the brain in coronal section and made confocal images in 14 brain targets. So here is just some of the example images, and you can see for the projection from the BLA to the nucleus accumbens, the densest projection is indeed in the nucleus accumbens, which is what we expected. And similarly, the densest projection for the neurons projecting to the central amygdala is the central amygdala, and the ventral hippocampus, the ventral hippocampus. So we then quantify the fluorescence intensity as I just described, and you can see that the reference is the highest in intensity for the 3 brain region. However, when you look at these data, there's something that was not consistent with what we expected from physiological data, which is a quite high expression of terminals or like of fluorescence in the central amygdala for the neurons projecting to the nucleus accumbens. And if you look at the relative axon density, it was reaching even 75%. So we wonder whether these were just passing fibers, which is one of the confound of this approach. So we decided to go for synaptic mapping. So for this approach, you still use a dual viral strategy, and you inject the retrograde CAF2-3 virus in the downstream region. And in the BLA of the upstream region, you inject a mix of the same virus, so that will label the axons, and a virus expressing synaptophysin M-cherry, which will be targeted to the synaptic contacts. And then you will see the EYFP axons and then boutons like the synaptophysin from the synapses. So with this approach, again, we use confocal imaging in the sections and quantify the relative density. So here at the top, we have the EYFP only, and at the bottom, we have the synaptophysin M-cherry. And you can see that for the central amygdala, when we looked only at the processes, you have a very high density, whereas when you look at the synaptic contacts, you have a much smaller collateralization. So this means that only 25% of these output are indeed synaptic, creating synapses in the central amygdala. So we did the same thing for the neurons projecting from the BLA to the central amygdala, and did not notice major differences between the axons and the synapses. Meaning this is also important to do this staining depending on the circuit that you're studying. Okay, so finally, for our last example, we are interested in mapping these projections in a whole brain. So I'm going to show 2 examples. So the first one is still focused on the BLA to nucleus accumbens projection. So again, we use this dual viral approach. In this case, we expressed only EYFP. And then we removed the lipids using the clarity approach that was developed by the Chang et al. In the Dysauth lab, and transferized the brain. So here is one hemisphere of the brain. And then we imaged the sample. So I'm going to try, oh, I have to remove the pointer, sorry, to play the video. Okay, so here you have the whole brain. This is the BLA, the anterior and posterior part. And you see the magnocellular neurons and the parvocellular neurons in the posterior part. And you can see and follow the axons going all the way to the nucleus accumbens. And what was very interesting in this sample is that you could see also the ventral path that was going from the BLA to the nucleus accumbens. Okay, so the clarity really allows you to follow the axonal path from the upstream region to your downstream target. So what you can also do using this approach is combine viral vectors, and you can look at different projection populations. So here in this experiment, we use the same approach with retrograde CAF2-3 to express M-cherry in the BLA to central amygdala neurons, and then we injected another recombinase, or another viral vector, retrograde viral vector, expressing another recombinase, which is FLIP. And then in the BLA, we injected two recombinase-dependent virus, so the AAV-CRE-DIO mCherry to label the BLA to CEM, and then the AAV-FLIP-DIO expressing the EYFP to label the BLA to nucleus accumbens. And then again, we went through lipid removal with Clarity and imaged the brain. So here is just a focus on the BLA, and you can see in green the neurons projecting to the nucleus accumbens, and pseudo-colored in cyan the neurons projecting to the central amygdala. So as you can notice, you can really see the dense dendritic arbor of the neurons projecting to the nucleus accumbens. You can follow the axons very well. However, for the mCherry mapping, you can see nicely the cell bodies, but the expression is not as high with the Clarity approach. So one advantage of Clarity compared to other transferization is that you don't need to do antibody staining. You can look at endogenous fluorescence. However, the efficacy for mCherry and EYFP is different. So maybe using other markers as a second fluorescent marker would be interesting. Okay, then the important point when you do whole brain imaging is and it's the bottleneck of this approaches, is the tracking and analysis of these fibers going from one region to the other. So there's one very nice study that has been published looking at these or quantifying these fibers, and they use an algorithm based on DTI analysis from humans, and they were able to map the projection from the MPFC to multiple downstream regions. And here you have an example of the projection isolated that go to the ventral tegmental area and the basolateral amygdala. So this is still like ongoing and in process. You don't have a software that you can just buy and use. It requires a lot of like computational skills and power also. You need a very big computer to run these analyses. Okay, so if I can give you take-home messages, I would say that first, you need to adapt your viral strategy specifically to your circuit. So try to read the literature if other people have already tried to map these circuits, and otherwise you need to test different serotypes and strategies. And why not combine with retrograde tracers? Then it's important to integrate collaterals when you do mapping of the selective projection. So as I've showed you, generally the main projection is the one you target, but you can have important collateralization. And it's also important to confirm that there's indeed synaptic contacts and it's not just passing fibers. Okay, so with that, I would like to thank you, and I'll give the floor to Nicholas.
Nicholas, you need to activate your...
My microphone. There we go.
Okay.
Hello, everyone. I'm Nick Foster, and I'm a scientist in the lab of Hongwei Dong at UCLA. We are a neuroanatomy lab, and so I'm going to talk to you today about how I select a tracer to use for a neuroanatomical tracing experiment. To do that, you need to know what the properties of the tracers are, and I like to not think of tracers really in terms of their good or bad tracers, but really, what are the properties of this tracer, and is that, most appropriate for the experiment that I want to do. So I'm going to explain some of the injection strategies that we use in our lab, and I'm also going to talk about a couple of the properties of some of these tracers to complement what Jose and Anna have already told you. One of the main techniques that we use in our lab is the double co-injection technique. So a co-injection is a mixture of an anterograde and a retrograde tracer delivered through a single pipette. So in this case, PHAL mixed with CTB or BDA mixed with Fluorogold. And that tells you the inputs and outputs from each injection site. And as neuroanatomists, we really like to use that technique because it maximizes the amount of information that you get out of each brain. Each of these brains takes a long time to allow the tracer to transport. Then you have to cut and stain and image it. So maximizing the data you get out of each brain is an important goal for us. But using double co-injections also has a secondary benefit in that it can reveal areas of indirect connectivity between injection sites. So for instance, if B projects to area X, which projects to area A double co-injection will reveal that. The anterograde labeling from B will terminate in X and overlap with retrograde labeling from A, allowing you to see this indirect connection between the two. This is an example of a double co-injection that we made. One co-injection is in the dorsal agranular insular cortex. The other co-injection is in the posterior agranular insular cortex. The first co-injection is fluorogold with BDA, and you can see that they are perfectly concentric. The second co-injection is PHAL mixed with CTB. And the CTB that we use is conjugated to Alexaflor 647. So it is naturally fluorescent in the far red. But the PHAL you have to stain for with the secondary antibody to be able to visualize that. And what I want to point out about this is that the injections are not perfectly co-localized here. Now, of course, they were delivered together. You can see the needle track here delivering both together. But the CTB injection is a nice ball in layer 5 here of cortex, whereas the PHAL labeled cells in green are present in layer 5 and in layer 6. And what happened is that PHAL is primarily taken up by the dendrites of neurons. And so these layer 6 cortical pyramidal cells have large apical dendrites that extend up through the injection site, and they absorb some of that PHAL and get labeled. So it's important to bear in mind, that this can happen with a co-injection, and that the labeling you see is going to reflect layer 5 for your CTB, but layers 5 and 6 for your PHAL. It's probably not going to matter, but it could matter depending on your application. Here is a sample of the labeling from that double co-injection. And you can see the retrograde labeling of CTB in the cortex here. There's some fluorogold in the amygdala here. PHAL looks great here in the thalamus, and BDA in the striatum here. In my hands, I've never been able to get BDA to work very well. This is about one of the best examples I could find. And you see the background is kind of higher, and the axons look a little more muddled compared to the PHAL. I think we have the opposite experience from Jose's experience. But this labeling from all 4 of these tracers is present throughout all 4 of these representative sections. I just showed one tracer for each of these sections for clarity's sake, but you get a beautiful brain-wide visualization of all of these tracers from a double co-injection. And you also see areas of indirect connectivity between the 2 injection sites. So in this case, the PHAL labeled axons from co-injection 2 in the posterior AI terminate here in the associative temporal cortical area, overlapping with fluorogold labeled cells from co-injection 1 in the AID. This double co-injection technique that I've been showing you has used all classic tracers. These are the chemical tracers. And there's a few properties of these, one of which is that they are non-selective, which means that all neurons or inputs to the injection site are going to be labeled. And as a neuroanatomist, I love this because I want to see all of the brain-wide connectivity with my area that I have injected. But I put all in quotes here because just like the viral tracers that Anna has talked about, the viral tracers have tropism issues potentially. The same thing can be true for some of the classic tracers. They may not label every pathway or every neuron type in your injection site area. So it's something to be aware of, and you probably want to try to use multiple retrograde tracers across, or intrograde tracers over different experiments if you have trouble labeling the particular pathway that you're interested in. As you saw, most of these tracers have really good signal to noise, which like you can see here in this PHAL labeled axons in the cortex here, which is important for creating good photomicrographs, good figures, and having a good baseline data set if you're going to do digital segmentation and reconstruction of your tracer labeling. And most of these tracers have both antrograde and retrograde activity. So as an example of that, I have this PHAL injection here in the caudoputamen, and the CP projects very densely to the substantia nigra pars reticulata. So you see this beautiful axonal terminal field here in the ventromedial SNR. But you also see this hazy labeling here in the substantia nigra pars compacta. That is retrograde labeling from PHAL. And how do I know that? Well, I put a good retrograde tracer, Fluorogold, in that same region of the CP, and I see beautifully labeled Fluorogold cells in that same region of the SNC. So this is what retrograde labeling of PHAL looks like. And usually it's a lot weaker. So, you know, these tracers are going to have both antrograde and retrograde, but they'll do one better than the other. And that applies to Fluorogold as well. So Fluorogold does have some antrograde labeling property as well, but it's hard to see. So I'm going to show you this same Fluorogold labeling image right here, but without the Nissl background so that you can see it a little bit better. And you see the good labeling in the SNC, but right here you see a little speckling of Fluorogold labeling. That is what antrograde Fluorogold labeling looks like, and it's in that same ventromedial corner of the SNR. So again, this isn't something that I would put into a figure, but it's a property of these tracers that can be potentially useful. Another way to use the double co-injection technique is to demonstrate interconnected networks of brain areas. And I recently used this to demonstrate the cortical basal ganglia thalamocortical loop, which is one of these classic canonical brain motifs. Cortex projects to striatum, projects to the substantia nigra, which projects back up to thalamus, in this case, the perifascicular thalamus, which closes the loop in a recursive manner back up in cortex. This network has been postulated since the 80s at least, but as I was looking at the literature, I realized that it had only been demonstrated in pieces, and all of those pieces had been put together to postulate this loop. This had never been demonstrated in a whole intact loop in a single animal, either anatomically or electrophysiologically. So I realized that demonstrating this loop could be possible using the double co-injection technique. So I put one co-injection into the CP and the other co-injection into the opposite node of this loop in the perifascicular thalamus. I targeted the ventrolateral part of the CP here and the ventrolateral part of the PF because I had done some preliminary experiments that indicated that these were networked together, forming one of these loops. In this case, rather than use BDA, I opted for AAV RFP because it works better in my hands than BDA does. But you can't mix AAVs with Fluorogold because the Fluorogold will kill the virus. So I had to inject these separately, but just targeting the same region. So you can see that the center of the Fluorogold injection is a little bit offset from the center of the AAV injection, but they mostly overlap. And I was able to use some of the connectivity labeling from these two injections to ensure that they are located pretty much in the same area. And we saw that even with like directly mixing PHAL and CTB, in that previous example I showed you, you don't always get perfect concentricity of your injections. Then I used CTB conjugated to Alexaflor 647 and AAV GFP. And in this case, I opted to use the AAV rather than PHAL for convenience sake, because at that point, PHAL would have been the only non-natively fluorescent tracer I was going to use. So I swapped it out for GFP, and that way all of my tracers are fluorescent. And that means I don't have to do any immunohistochemical staining, which saves me about a week's worth of histology time. So I can just cut the brain, do a quick 2-hour stain for Nissl, and then start imaging my slides right away. And what I expected from this was that if this cortical basal ganglia thalamocortical loop is an intact closed recursive loop, I would see overlap of the labeling in the cortex and in the SNR. And that's what I saw. So in the SNR, we see perfect overlap between the retrograde labeling from the PF thalamus in Florigold here and anterograde labeling from the striatum with these green labeled axons here. And then in the cortex, I found overlap of labeling in a cortical column in the primary motor cortex. So in purple, you see the retrogradely labeled corticostriatal cells, and in red, the anterogradely labeled thalamocortical axons here in layer 4. And while these don't technically directly overlap, a bunch of previous work has shown that cortical columns are integrated functional units, and the layers of a cortical column communicate with each other. But of course, I followed this up with a functional electrophysiological recording experiment to demonstrate that these axons do actually innervate the apical dendrites of these corticostriatal neurons here, showing that this is a closed recursive loop. Another technique that we use is the triple anterograde method. So we put 3 anterograde tracer injections, typically into a large brain area or a large brain region, or brain regions that have convergent connectivity to look at the topographic patterns. So in rostral CP, we put AAV RFP. In the intermediate CP, we put PHAL. And in the caudal CP, we put AAV GFP. And we can see the topography of striatal projections to the SNR have a medial lateral gradient. So rostral CP projects medially, caudal CP projects laterally, and intermediate CP is in between those two. The labeling from these is good, high quality labeling with good signal to noise. So we were able to digitally reconstruct some of these. And I'm going to show you the green labeling here on the next slide. This, we were able to digitally segment and reconstruct these axonal terminal fields. And doing that allows us to know exactly how many pixels of axon are present in the SNR and exactly where they fall, which is a huge benefit for doing computational analysis of the neuroanatomical connectivity. For doing retrograde tracing, we like to use rabies viruses. Rabies is a highly specific retrograde tracer. I've never seen anterograde labeling from rabies virus. As Jose explained, rabies typically has a polysynaptic infectivity. So you can get first order neurons and then second order neurons if you let the infection continue, and even third order neurons beyond that. But we use a variety of rabies called glycoprotein deleted rabies, which the glycoprotein is an element of the viral capsid. And so deleting that prevents it from spreading transsynaptically. So glycoprotein deleted rabies have a 1 synapse limit, which makes them work kind of like a classical retrograde tracer. And to visualize the infected neurons, there are a number of markers. They come in GFP, RFP varieties, as well as a bunch of spaghetti monster versions that you have to stain for immunohistochemically, but then you can put those in any color channel you want. So we can look at multiple rabies injections in a single brain. In this case, we made injections of GFP, RFP rabies, and a V5 spaghetti monster rabies. All of these injections are into different parts of the CP. And in this sagittal section, you can't see the injection sites, but you can see some of the labeled cells, the RFP and the V5 neurons here in the thalamus. And up here in the anterior cingulate cortex, you can see some of the green labeled neurons. And to me, that field of green labeled neurons looked really good. So I photographed that at high resolution with a 60x lens on our confocal microscope, and I obtained this image. And I love this image. I find it mesmerizing, and it illustrates just how well rabies labels neurons. You can see everything. The dendrites are really well labeled. You can even see dendritic spines on them. Axons are well labeled, although it's hard to pick out axons in this whole mess of neurites that you see here. But with labeling this good, you can reconstruct these neurons. And so for this final slide here, I just wanted to show how I used rabies to label neurons in the SNR. And because they're so well labeled, we were able to reconstruct a subset of these neurons to see part of their dendritic structure. And then I took that reconstruction image and overlaid it with reconstruction images of some of the striatal inputs to the substantia nigra at that level and layered them all together here. So you can see 4 terminal fields here reconstructed from injections in these 4 regions of the dorsomedial stratum here, colored blue, pink, tan, and gray. And putting this all together reveals some features of the functional architecture of the striatonigral pathway. So you can see that each of these axonal terminal fields from the striatum, they converge and overlap here in the ventromedial striatum, but they all each kind of have their own separate corner of this region that they're innervating as well. So you get partial overlap and partial kind of non-overlap of these terminal fields. And what the rabies labeled reconstructions show is that pretty much all of these neurons in this region look like their dendrites are extensive enough that they can contact all 4 of these terminal fields and integrate all 4 inputs. But by that same token, it looks to me as though, depending on where that neuron is within this region, it probably receives stronger input from 1 or 2 of these striatal inputs compared to the other 2. So it probably has a preferential innervation from 1 or 2 of these regions compared to the other 2. But it's possible that whatever particular spatiotemporal activation you get of these 4 inputs, you could potentially activate any of these neurons in here in a particular pattern. So that shows the functional architecture, at least gives some hypotheses for how the function of these pathways might work. So that's all I have for you today. This is our lab. That's me off to the side here. Thank you very much.
Okay, thanks a lot, Anna and Nicholas. So we're about to start the Q&A session. We have a number of questions in the chat. I just start to answer some of these questions, but I think that it's better to make a round trip and go through all of them. So first question we have in the chat is coming from Sadiq Adetayo. He's asking about what could be the effects of using green rabies virus as a guide for GCAM blind model. Well, Sadiq, as I mentioned to you before, I think rabies virus is not green in itself. So visualization of rabies virus just simply depends on the dye that is coupled to the secondary antibody you're about to use. So don't be that much concerned about green rabies virus. So green, so it's just a rabies virus that can be detected with a secondary antibody of your choice. So, and the second question is by Heather Brenthouse. I already replied to her, to Heather. She was asking about time course or survival times for all these tracers. And, there's a great variety of survival times. So there are tracers such as PHEL and BBA that can be detected anytime between 1 week up to 3 months. For fluorescent tracers such as chlorotoxin, fluorogon, survival time typically lasts for 1 week, maybe 2 weeks, probably not much more. For
I can just add for the CTVs, because I have used combination of viral vectors and CTVs. So I know that CTVs of different coupled to different fluorescent marker, you get up to 4 weeks or even 5 weeks, you can see labeling. So yeah, maybe it's not as good, but it's still working.
Yeah, to the very best of my knowledge, there's only a systematic paper being published in trying to address this question when dealing with PHEL. It's a very old paper by the Dutch guys of Hein Brennebergen and Floris Bouterlot. They made injection of PHEL in a number of animals, and they started to sacrifice these animals in different time course. So they found a nice PHEL signal with survival times of up to 3 months without any trouble. So at the end, it's something you have to do by yourself. Tracers that are no longer in use, such as Horseralis peroxidase, can only afford a survival time of less than 2 days. Otherwise, the peroxidase precipitate gets quickly metabolized and is no longer detectable. So next question, and Anna and Nicholas, feel free to answer any question at any time. This is a question by Cristina Miquelet Palomo, asking about that for patch clan experiments with long-lasting retrograde labeling, would you recommend as being the less toxic one for the neurons?
If I could chime in here, because I did
just go ahead.
Experiments like this. Yeah. I have used retrobeads as a retrograde tracer. So these are microscopic latex beads that are conjugated with rhodamine or fluorescein, but they do have a kind of toxic effect on the cell. I think if you're just doing tracing with them, you can let that tracing go for several weeks and you're okay. But for patch clamp, the neurons are really sensitive when you try to patch onto them. So you want the retrograde tracer to be in there for like 1 week max. So I had to end up doing 2 surgeries. I did 1 surgery to inject the channel rhodopsin, gave that a couple of weeks. And then I did a second surgery to inject the retro beads and gave that less than a week. It depends the distance that the retro beads have to travel, how long you want to let them incubate. But I found that 4 or 5 days was a good amount of time when I injected in the striatum to retrogradely label cortical neurons and get a good population of cortical retrograde labeling from those, whilst not having the toxic effect of the retrobeads affecting those neurons. I haven't tried other retrograde tracers.
So maybe I can pop in here.
Yes,
So
go ahead with your view.
The experiment I was just mentioning earlier is it was for patch clamp. So I used CTVs to combine with viral expression of channel rhodopsin URFP. And with the CTVs, the neurons were very healthy. So I was using the 647 to not overlap with the EYFP channel. And yeah, up to 4 weeks, I had like good labeling. I could patch the neurons that were healthy. So there's a bioarchive paper on that from 2015, 16 on food intake and amygdala projections. So the first author is Gwendoline Talun. But yeah, we did this combination of tracers and then patched the neurons labeled with the CTVs.
Well, I just want to add a comment that, Christina, it depends on what you are looking for. I mean, if you want just to have a morphological correlate of the neuron being patched, there are traditional alternatives such as biotin or neurobiotin that are very well tolerated by the neuron and spread very well within the patched neuron. But the trouble with neurobiotin and biotin is that they only last probably for less than 40 hours of survivable time. Otherwise, they will quickly metabolized. Okay, so let's move on to the next question by Heather Penhouse. Wonderful talk, wonderful and very helpful talk. Thank you too, Heather. I have a similar question about CAF3 intersectional approaches regarding time course. Could it be possible to do injections and then expect expressing tracing in, let's say, 10 days? So Anna, Nicholas, I think that this question is better suited for any of you.
Sorry, I was actually pasting the paper referring to what I was just saying, so I didn't follow. So the question was, sorry, regarding the CAF3 injection and the intersectional approaches, could it be possible to do injection and then expect expression tracing 10 days after? That's a little short amount of time, I would say. So it really depends also on your AAV serotype, but I think you need to try in your circuit, and it depends also on the density of your population. If you have many neurons, you can also try to use the retro AAV. That might be more efficient. Yeah, so I think if it was my experiment, let's say in an insular circuit, maybe I would go with if I had to go for a short time, I would do retro AAV and an AAV9 combination. But you need to try.
Nicholas, anything you want to add?
No, I agree with Anna. Okay. You can see labeling, in 10 days. It just may not be as bright as you want it to be. You can see it in less than that, but it may not be as bright as you want it to be. So typically we let pre-dependent labeling go 2 or even 3 weeks.
Okay, so the next question by Marta Nowakowska. Can you use the same virus for neuronal tracing in organotopic or atypic brains like cultures? I think they are making mention, Marta, to AAVs, perhaps. Well, my point is that, for cultures, AAVs are not the best virus to trace these neurons. Most people will rely on lentivirus instead of AAVs for cultures. But, I was wondering, Anna and Nicolas, you have any personal view on this regard?
I have no experience with the culture, but my concern would be more the time of expression, like for the virus. I don't know how long you can keep your culture, but yeah. I know there are publications using some, but yeah, I don't think it's AAVs, right?
I think most of them are lentis.
Yeah, lentivirus.
Yeah, I think that's
because they express much faster, and yeah.
Lentivirus for transducing cultures are much more powerful than AAVs. That's for sure. Okay, so next question is by Ole Borup. This directly to Anna. So what kind of microscope did you use for your tissue cleared hemisphere, Anna? Light microscope, blaze, size?
Yeah, so actually these images I've shown were taken in 2015, no, 17, 16, 17. And at that time we were using a confocal microscope. That's why we have such a good resolution of the dendrites. So yeah, today the way to go would be to use a light sheet microscope. We're currently exploring options to buy one, and indeed the blaze and the mesosperm are in our top choices. So yeah, they're both really good tools.
Okay, Anna, thanks a lot for your input. So next question by Koa Yumian. Hello, I'm a new engineer by training. I would like to learn more about tracing and calcium imaging. Can you point me to some textbook of reviews for an introduction and overview? Thank you. Well, if I remember properly, the webinar will soon be available in the FENS website. At the end of my talk, you have a couple of suggestions of review papers that can be used just to get started and be more familiar with the topic. And if you have any further question, feel free to drop a message and I will answer you without any trouble. Okay, so for the next question, Marta is asking us, can you also use those methods outside the brain, spinal cord or peripheral nervous system? Any of you, Anna or Nicholas, do you have any experience in this regard? You want to comment?
Yes, I have some. Yeah, you can use some of these methods in the spinal cord and even in the peripheral nervous system. But there are differences. These techniques that we've talked to you about, they all work, great in the brain. But for the spinal cord can be really hard to access. The cervical spine is easier to get to. But and the tracer techniques, you can make injections in there. But for the peripheral nervous system, I've had better success using the chemical tracers, retrograde tracers injected peripherally to label within the brain or spinal cord, in particular CTB and Florigold. And even then, it's been problematic. I've had to make large pressure injections. I typically like to use antiphoretic injections, but I injected large volumes, pressure injected, to see the labeling taken up from the peripheral terminals. And virally, at least in rodents, they don't work very well peripherally, in my experience. And I've seen some papers that have shown that if you want to use rabies peripherally, you need to inject the rabies in pups, less than, I think, P17 or something like that, to get labeling within the brain. Because once they're older, I guess something about the adult mouse's immune system is really good at cleaning up viruses, so they tend not to work very well. And I've made retrobeads injections as well peripherally, and I haven't gotten that to work either. I haven't done it extensively, but Anna, do you have anything to add to that?
No, during my PhD, I was working on the spinal cord, and we would use retrograde Alexa fluors that we would put on the nerve terminals, and it would be that would travel back to the motor neurons and sensory neurons. But yeah, and it was actually in spinal cords of frogs. So it was working, but that's my only experience. If that's interesting to you, I can actually share my PhD.
Okay, thank you. So let's move on. Next question by Nadia Seminara. She's asking about how do you quantify injection sites when using viral tracers? The reason I ask is that with traditional chemical tracers, you get a clear injection site, which makes analyzing the tracing between injections more straightforward. So, any of you?
Go ahead, Anna. Go ahead.
Well, yeah, I would say that the volume you inject of viral vectors is really important. So you need to inject small volumes. So I still see some papers with like more than 300 nanoliters of viral vectors. That's for mice. It's a really big volume. So try to keep it lower so you can see a defined region of expression. So I'm speaking of the anterograde viruses. So if you inject in the cell bodies, then we use confocal microscopy on slices, and we try to define the extent of the spread of the expression in all dimensions: anterior, posterior, mediolateral, and dorsoventral. And then we define the center of the injection across that. What we also look at is the actual needle track. So generally that will generate a little bit of gliosis that you can see at the tip of the injection site. And this is what we also use to identify the CAV2-CRE injection or the retrograde viral injection. You can also mix the CAV2-CRE with another viral vector or a tracer to see the injection site to help you visualize it. These are tricks that you can use. But yeah, generally, if you don't inject too much, it's reasonably feasible to define your injection site. That would be my answer.
Okay. So next question by Ingrid. I think she was asking about data shown by Nicholas. I'm not fully sure. The question is, which tool did you use to computationally extract and quantify axonal labeling?
Good question, Ingrid. Yes, we developed software in our lab to do this. Our lab has both the wet lab component, with neuroanatomists like me, and a dry lab component with computer scientists and programmers that developed this tool for us to use. But even before I joined this lab, I was using Photoshop to do stuff like this. So if you get a good quality photomicrograph, export it as a TIFF, and if it has good signal to noise, put it into Photoshop. And Photoshop has a bunch of great tools, especially the extended edition of Photoshop for segmenting the image. And I think with the extended edition, you can even use it to count the number of pixels in an area that you select.
Okay, thanks a lot, Nicholas. Next question by Lara Kirik. She's asking about when we are doing double quantitative labeling, which control we can use to know the tracing efficient. It is possible to count how many cells are receiving projections. If the efficient is not high enough, maybe we are counting as negative cells that are part of the path, but they just were not tagged for some methodological reason. So, Nicolas and Anna, do you want to comment on this regard?
Well, I can comment. Yeah, obviously you have differences of efficacy between the tracers, and you will never get all if you use a retrograde tracer, for example, you will never label all the neurons that go to that site of injection. So you will always have to include that non-labeled neurons might also project to your region of interest. So this is inherent caveat of the experimental design. So I think, yeah, there is no real way around that. Even if you use molecular markers or transgenic lines, if there is a viral injection or tracer injection, it will not have 100% efficacy. So.
Okay, well, just, Laura, as a comment on my own experience, my best advice, is that, of course, there are different AV cell types that perform in different ways, as Anna has shown before. So my best advice would be not to rely on just one technique. So if you are using, let's say, AVs for retrograde tracing purposes, you need to duplicate the experiment with, let's say, a chemical retrograde tracer, just to make sure that, it's not a matter of efficiency in terms of retrograde transport. So don't put all your outputs in the same cage. So just try to reproduce experiments by using tracers of different nature, not only just one type of tracer. Nicholas, anything to comment on your side?
No, nothing else to add.
Okay, so let's move to the next question by, Pilar Martinez Olondo. Thanks for the great talks. They were very helpful. The question is, when would you choose rabies tracing better than other retrograde tracers such as TTB?
Well, rabies, like I showed, is great for labeling the whole neuron, including the dendrites. Some of with the chemical tracers, with the retrograde tracers, you can see the soma and maybe some of the proximal dendrite. But when you have axons coming in, they may be synapsing on some of the distal dendrites. So in that case, you might want to opt for using rabies as opposed to CTB or Florigold to be able to better visualize potential synaptic contexts in that situation.
Yes, I cannot agree more, Pilar. The point is, choosing either CTBs or rabies depends on what you want to look at. So you are really interested in visualizing the entire dendritic arborization together with dendritic spines. I think it is without doubt that using Rabies virus will give you a much better image in terms of intracellular feeling of the neuron to the best possible morphological detail. If you just want to count cell bodies just to quantify, let's say, number of neurons giving rise to a given projection, I think that using CTB is much more straightforward. So basically, it depends on what you want to see. Okay, let's move to the next question. Lasani is asking about, do you have experience in performing in vivo patch clamp in an area where you've done viral injection? Is there any difficulty patching this due to damage associated with virus injection? I never tried this before.
On my side, no. On my side, no. For the viral injection, I never had problems, like even if it was around really close to the injection. I have not done that personally, but I have heard from people mentioning that if you try to patch below an optic fiber implant for optogenetics or calcium imaging, that it might be harder to patch, but for the viral injection, I never noticed damaged neurons.
I didn't either. In the few experiments that I've done that way, I think it might be possible if you do a large pressure injection that there could be some damage in the area, but I did interferotic injections.
Okay, next question by Julia Torromino. She's asking us about, do you have any suggestion for multisynaptic anterograde tracing from the spinal cord? Is the spinal cord different than the encephalon in your experience? Julia, on my side, I never tried to make injections of anterograde traces in the spinal cord. Normally, these are GHL or PDA. But, as far as I understand, they are going to work more or less exactly the same way as in the brain. Nicolas and Anna, if you want to comment, feel free to go ahead.
Just about the multisynaptic anterograde tracing. I know there's some publication of some hybrid serotype of AAV that would have polysynaptic anterograde properties. I have not tried them myself, but I think it's worth a look if you're interested in that. Okay, so you don't have anything more to comment. Let's go to the final question that is made by Patrice Fort. Is that possible to inject AAVs by macroiontophoresis as classically done for CTB, neurobioptin, PHGL, or Flowbone in order to get smaller injection sites? Yes, definitely. And I can use even smaller micropipette tips with the AAVs than I can with the classical chemical tracers. Okay. Well, there's one more question coming from Sadiq. I'm not sure if I understand this correctly. Jose, do you mean rabies virus will be helpful in identifying the exact number of neurons in a volume imaging, for instance, for example? Well, I think that rabies virus is doing a very good job. So for sure, I think you can identify the exact number of neurons. It depends how you are going to count these neurons. But there are a number of software, pieces of software based on artificial intelligence where you can train an algorithm
and to start counting neurons in a very accurate way. Nicholas and Anna, you want to comment?
I think I would just say again that, I mean, maybe I'm wrong, but I don't think that if you do a viral injection, even with rabies, you will transduce all the neurons. So if you really want to have a real count of the neurons, I would use like old school DAPI or Nissl staining, just to be sure, like, you know, yeah. It depends on your question. Yeah. Go ahead.
Possibly a transgenic line, you know, it,
yeah, also
could be used as well. But I haven't done stereoscopic counting of that kind before.
Okay.
I think it's also like if you use transgenic line, you will have like the whole arborization and you know, dendrites, you know, especially if you want to use computational tools or automated tools to do the cell counting. When you have the dendrites, it's really hard for an algorithm or software to separate the cell bodies from a big dendrite, for example. So yeah, I actually would not use that approach if you just want to count cells.
Mm-hmm. Yeah.
Okay, so if there are not any more questions, we are about to close the webinar now. We are clearly beyond schedule, but you know, we at the beginning we have experienced some technical difficulties. It is expected that this webinar will soon be made available in the FENS website. And you know, if you have any further question, please drop us a message, and we will try to answer as soon as possible. I just want to finish by you know, saying thanks to Nicholas and to Anna for your contribution. It was a great pleasure having you on board. And looking forward to meet with you in any forthcoming meeting, who knows where. So have a good day, enjoy your weekend. Thanks a lot to all attendees for joining us to this seminar. Thanks a lot, and bye.
Thank you all. Bye.
Thank you. Bye-bye.
Bye.